Recording in progress.
Ladies and gentlemen, esteemed guests and fellow conservation enthusiasts, we would like to welcome you back from your lunch. We hope you had a very pleasant break and lunch. Welcome to the International Symposium on Wildlife, Biodiversity and Conservation. Before we start, we would like to introduce ourselves. My name is Sabila Nabihati, and beside me is Rizka Afif. We are here as the Master of Ceremony for this symposium. Before we introduce our chairperson, we would like to take a moment to acknowledge and express our gratitude to the organization and sponsors that have made this event possible. Their unwavering support and dedication to the cause of wildlife biodiversity conservation have been instrumental in bringing us all here today. Let us recognize
the organizer, Faculty of Forestry at Universitas Gadjah Mada,
Ministry of Environment and Forestry,
and there are also our sponsor from April Group,
Global Environment Facility in partnership with United Nations Development Programme and Combating Illegal Wildlife Trade,
Peat and Mangrove Restoration Agency of the Republic of Indonesia,
Borneo Nature Foundation,
Taprobakina, the Journal of ASEAN Biodiversity,
Flora and Fauna International,
Copenhagen Zoo,
Kaleka,
Action Conservation Foundation,
and Pesona Indonesia Tours.
We are deeply grateful for their commitments to advancing our shared mission of conserving and protecting in the incredible wealth of biodiversity of our planet possesses. Now, without any further ado, it is my privilege to introduce our chairperson of this symposium, a respected leader in the field of conservation, who will expertly guide us through the session and discussion to come. Please join me in welcoming our chairperson here, Dr. Francisca, who will take the reins from here and lead us on this enlightening journey of exploration and collaboration. Before, let's make a noise here and give the applause to Dr. Francisca. All right, Dr. Francisca, time and screen is yours.
Thank you very much to the MCs who are very lovely. Welcome to Symposia 1. Today we have 8 presentations, so this is abstract presentations. Each presenters will have a maximum of 15 minutes, and I would like to give the freedom to presenters however they would like to arrange their time. So 15 minutes in total. You are welcome to speak for 10 minutes, for example, and then follow with 5 minutes Q&A, or how is it you prefer. We have 8 presenters. Some will be joining online, and I think most of us will be presenting from here, from this room, yeah? I will be assisted by the organizing committee. There are MCs here and note takers in the back, I think. These presentations will be broadcasted from the committee as well. We have the complete set of presentations. Good. Yeah, I think that's all. Any questions or comments before we start the symposia? Right. And also welcome to the online participants. Today we have 37 participants joining us online from, I believe it's from across the world. So may I present the first speaker today, which is Dr. Mark Harrison. I see Mark is already here. Hi Mark, how are you?
I'm good, thank you.
Mark Harrison will give a talk with the title of Impacts of Tropical Peatland Protection and Restoration on Tree Biomass and Species Richness in Sebangau National Park, Central Kalimantan, Indonesia. So can we see Mark here in this room, please?
Do you want me to share my screen or will it be displayed?
Can we pin Mark on the screen, if that's okay?
Do you want me to share my screen?
Yeah, okay.
Can you in fact hear me?
Mark, can you let us know if we can hear your audio, okay?
I can hear you, yes. Can you hear me?
We cannot hear you yet.
Ah, okay.
Mark, once again, if you would like to try speaking so that we can see if we can hear you.
Yeah, sure. Okay.
Good.
I think you can hear me now?
Yep, all good. Will you be presenting or the presentation will be from us?
Yeah, I can present. If you want me to share my screen, that's okay. I can do that. Or if you put up on your screen, I don't know.
Committee, do we have? Okay, so we'll share your presentation from here. So let us know when to move on.
Okay, perfect. Thank you.
Okay.
Okay, are we ready? I can't see the presentation anywhere.
Yes, as soon as we see the presentation, you can start.
Okay, great.
Good morning, by the way. How is it in the UK?
Thank you. Yeah, it's good. Thank you. Yes, thank you very much. A little bit early here, but yeah, it's great to join. Thank you for thank you to you and to the committee for allowing me to join online today. I'm very happy to be here. Sad I can't be in the room with you, of course, but yes, happy to still be able to join and still be able to contribute to the meeting today.
Okay, perfect. Yeah, the time is yours, Mark.
Okay, great. I can see. Hold on just a minute. Okay. Okay, yes, great. Well, yeah, thank you very much. Yeah, today I'm going to talk about some of the impacts of tropical peatland restoration, protection and restoration on trees in Sebangau National Park in central Kalimantan. Yeah, if we could go to the next slide, please. Next. Okay, great. Next. Worldwide, most forests have actually experienced some kind of human disturbance now. There are hardly any forests anywhere in the whole world that haven't experienced some kind of human footprint. And because of that, and because many of these forests are quite heavily degraded nowadays, forest restoration is a major international and national conservation priority. And this includes both assisted regeneration, where tree species are planted in degraded areas, and also natural regeneration, where forest areas are protected so that the forest can recover through its own natural processes. And the impacts of logging disturbance on forest structure and biodiversity have been fairly widely studied across the world. But recovery from these disturbances has been maybe less well documented. And that's particularly so in terms of long-term records from the same site, as composed to spatial comparisons between different areas. And Indonesia represents a good a good case study for this, because many tropical peat swamp forests in Indonesia were previously allocated as logging concessions or experienced past logging, but are currently being protected. And so there is currently a great opportunity here to study the recovery of these forests. Next, please. Okay. And so in relation to that then, some of the key questions that we asked here were, what are the impacts of past logging disturbance on tropical peat swamp forest structure and tree species? Secondly, what is the potential for recovery of these after disturbance and following protection of the area? And as a final question, what might this mean for wildlife? Next, please. And we conducted our study in the special research zone of the Sebangau National Park in central Kalimantan. And this is an area that is well known for having quite high diversity of flora and fauna, with many globally threatened species living there, and many legally protected species in Indonesia as well. So a very high priority conservation site. Also thought to be home to the largest protected population of the Bornean orangutan. Next, please. And in this site, we're somewhat fortunate because there has been a long history of. Oh, someone's saying they can't see the presentation on the screen from the comments there. I don't know if that means people.
No, I think it's signal in their location, because I on Zoom and I can see your presentation well.
Okay, all right. In which case, I will just carry on.
Please.
Okay. Yes. So as I was saying, we're lucky in this site because there has been a long history of research here. And this includes from before the national park was created. So before the national park was created, there was a logging concession in this particular site. And following that, there was then when that logging concession ended, there was a period of illegal logging that ensued. And that then stopped in 2004 when the national park was created. And since then, the area has been protected, and so with lots of restoration efforts, peat re-wetting, tree planting, forest patrols, etc. And so potential there for the forest to recover from that past disturbance. And the first tree plots in the area were established in 1993. And since then, there has been regular monitoring of tree plots in the area, so we can actually assess patterns within trees over time. Next, please. Yeah, thanks. And so this has been done through monitoring tree plots, within which all of the tree species over particular size limits are on every time they're checked again. These plots, the trees are categorized as being either alive or dead. And the basal diameter at breast height are measured, and the trees are all identified to species level where possible as well. There's actually two different data sets included here. The first was collected by a different set of researchers in 1993. So this used 3 plots that measured 50 by 30 meters and only included trees that were more than 7 centimeters diameter at breast height. And the second data set, the main data set, if you like, was initiated in 2003, and that includes 6 different plots with 2 different size limits of trees. So the first smaller plots, 5 by 300 meters, include trees that are over 6 centimeters diameter of breast height, and some slightly larger plots, 5 by 500 meters, include trees that are over 20 centimeters DBH. And these plots have been measured every 2 years. And from these measurements, we can establish the density of tree stems, and also the basal area or cross-sectional area of trees within each plot. And this map here just illustrates where the plots are. You can see those kind of white bars, which are which are the 6 plots used there. And from these measurements, we can subsequently also calculate tree biomass, and from that also tree carbon stocks using allometric formulae. Okay, next please. So moving on then to some of our findings. If we look here firstly at stem density, and this here is restricted to just trees that are over 7 centimeters dBH in the different years. The color codes that are being used on these bars are consistent between the different charts that I will show here. So the yellow bar, sorry, for 1993 was during the logging concession era. The red bar in 2003 was during the period of illegal logging, and green after that is following the creation of the national park and all the protection efforts associated with that. So what we can see there, if we look at stem density of the smaller trees, is that there is an initial increase following the logging period, and then it sort of goes down somewhat after that. Next, please. But if we look at larger trees, we see a slightly different pattern. So these are the trees that are over 20 centimeters dBH, and those increase consistently throughout the monitoring time period. Next, please. Moving on to look at the total basal area, the total area of tree per hectare of plot. And again, we see more similar patterns really between the two different size classes of trees, between the smaller trees and the larger trees. Just saying, the larger tree measurements we don't actually have available from 1993, so those are only from 2003 at the moment. Hence why there's only one bar for 1993 there. But for the total basal area, we see an increase for smaller trees and then a stabilization from about 2013. And for the larger trees, we see a more consistent increase throughout the period. Next, please. Total biomass shows a roughly similar pattern that there increases for both tree sizes throughout the period. Next, please. Next, please. Okay, great. And unsurprisingly, because these things are calculated from the same metrics, essentially, there's the same pattern there for tree carbon stock as well, increasing throughout the period. This is just for trees that are more than 7 centimeters dBH. Next, please. And here, looking at species richness, so this is a slightly different pattern. This is here we're looking at the total number of tree species found across all of the different plots. So bear in mind here that the plot size was different for the larger trees than the smaller trees. So these figures aren't directly comparable between the two different size classes of tree here, but they are comparable within themselves, so they're comparable for assessing the trends over time. And here, if we look at the smaller trees, we see basically roughly stable tree species composition for trees less than 6 centimeters dBH, with a slight drop towards the end, towards the last couple of measurement periods. For the kind of medium-sized trees of around 10 centimeters dBH, the number of tree species are very stable over time. In fact, they're exactly the same number. I think that was 97 species in both 2003 and 2021. But for the larger trees, we see a small, but nevertheless a consistent increase throughout the period there in terms of number of large tree species. Next, please. Okay, so I presented a number of different graphs there, which hopefully weren't too confusing, but just to sort of summarize what those showed, I'm splitting here into the small trees, 6-7 centimeters dBH, and the larger trees over 20 centimeters dBH. So for the small trees, then we saw an increase and then a slight decrease in tree density. For basal area, we saw an increase and then stabilization. Biomass and carbon stock increased over time, and species richness was stable and then decreased very slightly. But for large trees, we saw a slightly different pattern, and they increased in all metrics throughout the period. Next, please. Okay, so what does this mean? Well, generally, this seems to present a picture of forest recovery and tree growth over this period with protection that was that ensued. And we do see some decreases in some of the metrics for the small trees, but that is actually really to be expected as the small trees grow and become larger trees. So the metrics for the larger trees increase as some of the metrics for the smaller trees might decrease correspondingly. And the basal area of trees actually now exceeds the pre-illegal logging levels for trees of 7 centimeters dBH. And so this is this looks like a very positive picture overall here, really. And it seems particularly important that the basal area, the biomass of large trees and the species richness of large trees are increasing, because large trees produce the most fruit and are thus most important for forest frugivores, so for species such as the orangutan. And as I say, this seems to indicate natural regeneration has been occurring in the site following past logging disturbance, and so suggests positive impacts of peat swamp forest protection efforts in the area. And just a couple of little caveats to that. These are some preliminary analyses, and there are some differences in terms of plots measured between the earliest data set and the later data sets. Some of the estimates are still a bit crude at this stage. And there were some different botanist used throughout the period as well, which might affect some of the species counts. And from an ecological perspective as well, there has been the history of peat drainage and re-wetting of the site. So that may have also had some impact in terms of tree growth. Okay, next please. And yeah, those are just some of the references cited there next. And thank you very much to everybody for listening and for having me here.
Thank you so much to Mark. Let's give a round of applause for the excellent presentations. Very interesting and a very positive note as well, yeah, showing that the peat swamp forest is regenerating and restoring with efforts from human. Any questions or thoughts from the participants? I see one. Maybe we can take two. One from offline, and maybe if someone from online would like to also ask some question, I can read it from the chat later.
Langsung ya. Yeah. Great presentation, Mark. I'm Ardian from DICE. I have two questions. One is like 2015, we have like a very big fire. I'm just wondering if that affects your data. And the second one is quite a long period, more than 20 years. I'm just wondering if climate change somehow affecting the pattern of the growth of the trees that you are surveying in Sebangau National Park. So.
Would you like
yes,
to respond to that, please?
Yes, good questions. Thank you for those. Yes, as you say, there were big fires in central Kalimantan in 2015. These forest plots didn't experience any fire, so they were in unburned forest. So the trees themselves in those plots haven't experienced any burning. Nevertheless, yes, as you also say, there may be some effect of differences in climate and differences in rainfall, etc., between the different years, which would surely have influenced tree growth between the different time periods. Exactly how that has influenced tree growth, I don't think I'm able to say really at the moment. But certainly it might be expected to, and as I understand, there are projections of changing climate, potentially maybe even drier climate, I think, as a result of global climate change. So that is also likely to influence tree growth in the future. But yeah, we will have to hopefully be able to continue doing monitoring over the next decades, and hopefully we will be able to answer that question in future.
Thank you. He gives a thumbs up. Thank you, Mark, for the presentation and discussion. Let's give a final round of applause to Mark Harrison for his talk. Thank you. We'll move on to our next presenter, who are here offline, I believe. Let me check my schedule. Our second speaker is Dr. Dolly Priatna from Universitas Pakuan, who will be presenting a presentation entitled Behaviour and Home Range of Javan Slow Loris in Gunung Sawal Nature Reserve, West Java, Indonesia. Pak Dolly, dipersilahkan.
Thank you, moderator. So, good afternoon, everyone. Assalamualaikum warahmatullahi wabarakatuh. Is my presentation there? Oh, dah, baik. Thank you. So, distinguished ladies and gentlemen, all participants, we would like to share the result of our study about behavior and home range of Javan slow loris in Gunung Sawal Nature Reserve. So actually this is a part of my student thesis. So it was written a couple years ago. So probably the data used also not so sufficient, yeah. So as a preliminary funding. So next, please. So as we are aware that Indonesia has about 60 out of around 250 known species of primate worldwide, and at least 36 species or 60% of those of the total are endemic to Indonesia. So slow loris is one of known primate occur in Indonesia. So next, please. So slow loris distributed in South and Southeast Asia, including Indonesian Sumatra, Kalimantan, and Java Islands. And slow loris, actually slow loris is a little studied nocturnal primate native to Java Island, whereas actually their behavioral data could potentially aid the conservation management of the species. Next, please. So next, this is just the status and regulation. So the objective of this study is to determine the differences in daily behavior and home range of male and female rehabilitated Javan slow loris in Gunung Sawal Nature Reserve in Ciamis, West Java, Indonesia. So the information generated from this research hopefully can be used to develop and improve future Javan slow loris conservation efforts. Next, please. So this is the study area. Yeah. So this research was conducted in Gunung Sawal Nature Reserve. So administratively located in Tanjung Sari Village in Ciamis Regency in West Java, Indonesia. So as I mentioned earlier, that the field data was collected for only 6 months in 2018. So for Javan slow loris, comprising 2 adult male and 2 adult females, used as a subject of the research. So the 4 animals were either confiscated slow loris or had been kept as a pet by the community, and then they are rehabilitated in International Animal Rescue Rehabilitation Center. So the 4 rehabilitated animals were returned and then to Gunung Sawal or translocated to Gunung Sawal Wildlife Nature Reserve by the Natural Resources Conservation Agency of West Java in mid-2017. So just a year before the research was conducted. Next, please. So this study employed biotrack radio transmitter fitted to 4 individuals, rehabilitated slow loris, and in addition, we use a CSI receiver and a biotrack antenna to aid in locating the position of the slow loris, which have been fitted with a radio transmitter. Next, please. So this is the result. Yeah. So this graph is actually showing the percentage of daily behavior of male and female rehabilitant slow loris. So we can see from this graph actually their time budget, yeah, mostly used for traveling, feeding, and foraging. Next, please. So this graph also shows the percentage of use of vertical home range of the male and female. This is the activities, yeah, based on time segment. So as we are aware that the slow loris is a nocturnal primates, so they active actually mostly in between 9 p.m. to 12 midnight. Next, please. So this is the vertical home range. So they both the female and female use in between 0 to 10 meter heights within the canopy. Next, please. So this is using the MCP or minimum convex polygon method. Male Javan slow loris has a home range of about 46.9 hectare, while female has a home range 42.7 hectare. So while using kernel density estimator or KDE, so male slow loris has a fixed home range of 25.4 hectares, and the core of the range is only around 4.9 hectares. While the female slow loris had a fixed home range 20.4 hectares, and the core home range is 4.4 hectares. Next, please. So using the MCP or minimum convex polygon, the male Javan slow loris has a home range 46.9, yeah, earlier. So, and yeah, next please, sorry, that is horizontal. So the habitat preferences of male and female. So there is a 3 category of the habitat. So the plantations means an area belong to perhutani. So this area is dominated by pine trees and rasamala, all things, that's the hardwood in famous hardwood in West Java. So while that's the perhutani area, while a plantation is belong the community plantations. So while the wildlife reserve, so they use actually the female, most of these animals actually mostly use perhutani area. So even the perhutani area is dominated by pines or pinus, yeah, pinus trees and rasamala, but in the second layer they have like aren and kaliandra. So that's that area is very rich of food sources for them. Next, please. So as a conclusion that there is no statistically significant difference in daily behavior between the male and female Javan slow loris in Gunung Sawal Nature Reserve. But actually, theoretically, there should be differences between male and female in daily behavior, but because as I mentioned at the beginning that the sample use at that time is very small and the field data collection period is very short. So hopefully we can use the other data to combine with the previous data to gather the more comprehensive results. So based on the statistical test also there is a difference in vertical and horizontal range between male and female rehabilitant Javan slow loris. And the male Javan slow loris travel mostly at around 0 to 5 meters height, 58%, and female slow loris mainly at the height between 5 and 10 meters above ground. So the horizontal range on the male Javan slow loris earlier mentioned that 46.9, while the female slow loris is only 42.7 hectare. So in contrast, using kernel density estimation, so the male Javan slow loris has a home range 25.4 hectare and the female 20.4 hectare, while the core almost the same, yeah, with 4.9 and 4.4 hectares. Thank you very much. Next, please. Yeah, terima kasih. So return to moderator. Thank you.
Thank you to Pak Dolly. Let's give a round of applause. Very interesting talk about behavior ecology, and I myself coming from a rehabilitation background, so it's very related to this issue. Anybody would like to comment or ask some questions about slow loris? How about from the online participant? Yeah, I have someone. Oh, sorry, sorry. Okay, I'll go to Mr. here in person first and then come to the next question.
Okay, thank you. Just curious, Pak Dolly, as the result of the observation, 47% slow loris mostly in perhutani forest, right? And then 45% community and 7.6% in protected forest. So maybe I just want to know how about the effect of this research. Maybe there is some conflict, maybe Dolly, from the perhutani company or the community area in the close with the national protected forest, maybe because but to my knowledge that this species may be not as nocturnal, right? Nocturnal species. So there is some conflict for the community or the company. Maybe that's the first question. And the second, I just curious also, Pak Dolly, because the male mostly the travel area in the height of the trees 0 to 5 meter and the female 5 to 10 meter. So just my hypothesis, is the female species, they travel with the 5 until 10 meter, is it maybe make them safe from the predator maybe, or some of the predator or any more can disturb this female species? Maybe that's all, Pak Dolly. Thank you.
Thank you, Pak Saiful. So yeah, actually, as we are aware that the rehabilitated or translocated animals is still mysterious. Yeah, so I work a lot in Sumatran tigers. So even they are translocated far away from human settlements in the deep of the forest, so they tend to return to the edge in between forest and open area, yeah. So ecotone, so mostly with the village. So in tiger perspectives, so this is because of a lot of ungulates animal use that particular ecotone area. So that's why one of the reason tiger return to that areas. So back to slow loris. So actually, according to our observation on the ground, that the area in the perhutani area, yeah, in perhutani, even they are dominated by pines and rasamala trees, so they are in the second layer of the forest there. So actually a lot of food sources such as aren, and also in Sundanese called harendong, melastomase, harendong. So the smaller trees, so they eat the sap, getah, sap, and fruits as well as nectar from that trees. So that's why they travel so low. I think for foraging behavior, there will be not different between male and female, but in terms of other purposes, such as occupying the home range, that the male should have bigger areas than female. So in terms of foraging, they going down, supposed to be no differences in foraging for looking for food. So they very depends on the situation of vegetation there. As in this case, in the Gunung Sawal area in the Perhutani, so a lot of very low vegetation that provide food for them. So yeah, that's actually our assumption. Thank you, Pak Saiful.
Thank you, Pak Saiful and Pak Dolly. I'm afraid the time is limited, so I will move on to the next speaker. Thank you to Pak Dolly. Let's give a round of applause. There are still questions and comments on online Zoom chat. So if I may kindly ask the speakers to either join on Zoom and reply in the Zoom chat, or go to the committee in the back and reply the questions by Zoom chat later on, if that's okay with the speakers, Pak Dolly and the next speakers. Thank you very much. So we'll move on to the third speaker. Reza Pahlevi. Do we have Mr. Reza Pahlevi in this room? Do we have him online? Online. Oh, online. Okay. Reza Pahlevi, are you there?
Yeah.
Good.
It's my sound clear?
Yes, we can hear you loud and clear. If you can just open your video so that we can have an interactive.
Wait.
And we already have your presentation on our screen.
Oh, can I share screen because some different with
Okay.
the.
Panitia, bisa? Okay. Yes, go ahead.
I'm not permitted by the. Okay, wait. Okay, thanks for the time, and this is we will discuss about the presentation about change in the community structure of mangrove vegetation along the north coast of Mojo, Pemalang in Central Java post-rehabilitation. And we have some person, as we know that I am Muhammad Reza Pahlevi, and then Prof. Erni, and Ibu Niputu Diana, and Akmal Rujiat, and Mas Rian Adisatria. So, as we know that we have a lot of mangrove forests in Indonesia, yeah, and we know all of us mangrove forests are a type of ecosystem with very unique, yeah, with a very unique characteristic in tropical areas. And Indonesia has mangrove 3 million hectare, and 100,000 hectare is critical, and we must to be fought to maintain this sustainability. This data is from KLHK 2019. So the main problem in mangrove in Mojo is the mangrove area was designated as essential ecosystem area, or we call KEA, based on Pemalang Region's decree number 188.4/564/2017, date June 16, 2017, with an area of 14.5 hectare. And also based on geospatial data from the interpretation of digital global satellite imagery, the mangrove forest area of Mojo village in 2015, 16, and 17 has decreased every year. Yeah. In the maybe in 2015, the mangrove area was 13.52 hectare, and in 2016 it was 11.37 hectare, and in 2017 it was 8.62 hectare. So every time and every year the mangrove is decreased. And also the mangrove vegetation ecosystem in Mojo village is being harmed. The lack of public awareness regarding the conservation of mangrove area include unmeasurable use. And the people still use the fish farming, or we call as silvofishery in this area, is increasing in popularity. So this is the research mangrove in Indonesia. We have Purwanto in 2014 in Cilacap, and Puji Rahayu in 2017 in Pemalang, and Syamsu in 2018 in Asis area Surabaya, and Hilmi 2020 in Cilacap, and we don't know about in 2022. So I choose this topic. So this is the research objective, is to identifying the vegetation composition and the structure, the structure of mangrove in 2022 in Mojo district, Pemalang, Central Java. So this is the location of Mojo in Pemalang, Pemalang Central Java, in Central Java, in the North Sea. We have the characteristic of mangrove during the April until October 2022.
Excuse me, Pak Reza.
Yeah.
You have 5 minutes left for the presentation.
Sorry.
Thank you.
Okay. This is the result. We can see in Mojo, it's very different. Yeah, it's very different. You can see at the river, at the canal, we can see the differences about two. And this is the plot we have. So this is the individual number of mangrove species. I have Rhizophora mucronata as the highest number of individu with 51 species. And this is the seedling. We have Avritania alba is in the highest, and sapling we have Rhizophora mucronata, and the tree of growth rate we have Rhizophora mucronata also. So this is the density and relative. We have seedling, sapling, and tree. We have Avritania alba, Rhizophora mucronata, and the two if is the density in mangrove Mojo. This is the frequency. We can see the highest is Rhizophora mucronata and also Avritania alba. And this is the dominance and relative dominance. We have Rhizophora mucronata, and this is the INP, or index nilai penting. We have Rhizophora mucronata is the highest INP, and the lowest is we have Serioptagal. So this is the vegetation structure based by diameter. And the conclusion, there are 8 types of mangrove that grow in Mojo Village, Comal district, Pemalang. There are Rhizophora apiculata, Rhizophora mucronata, Avritania alba, Avritania marina, Soneratia casulariis, Casuarina escolitia, Bugera sexanua, and Serioptagal. And the structure of mangrove vegetation in Mojo Village, Comal district, Pemalang, we can see Java at the seedling level is dominated by Avritania alba. Meanwhile, the level of sapling and trees is dominated by Rhizopora mucronata. And the last is the mojo Pemalang mangrove ecosystem area, Central Java change in the structure of mangrove vegetation have occurred, which have resulted in a change in vegetation composition and damage to part of the area. So for the research is needed regarding area management and its yes. Okay, I think enough for my presentation. Back to moderator.
Thank you very much, Pak Reza. Let's give a round of applause to Pak Reza for his presentation about vegetation in mangrove. Any question or comment from this research project? Anyone? Okay, I'll move on to the next speaker. And again, if anybody will still have questions or comments, especially from online participants, you are more than welcome to put it down on the Zoom chat so that we can forward it to the speakers and let them respond at their earliest convenience. Thank you very much. We'll move on to the fourth speaker of this symposia. And it is Ms. Latifa Nuraini with the title of Conservation of Tropical Wild Orchids to Support Green Economy and Climate Resilience. Do you have Ibu Latifa here? Yeah. Silahkan Ibu Latifa. And the presentation, please, committee. And the time is yours.
Well, all right, thank you. So, hello everyone. Please introduce myself. My name is Nora from Research Center for Conservation, Plant and Botanical Garden Forestry, BRIN. So, today on behalf of our team, I would like to present our research entitled Conservation of Tropical Wild Orchid to Support Green Economy and Climate Resilience. So for the introduction or overview why we do this research, actually we conduct a research project funded by ASEAN and then for conservation of tropical wild orchid in Kars Mountain to support or empower local farmer. But we want to know about this Sleman City, so we conduct this research. So as we all know that Indonesia is a tropical country that rich in germplasm, and one of it is Orchidaceae, or we call it orchid, and there are many, around 5,000 species, and that the tropical orchid are often threatened with extinction due to the land clearing for settlement, for example, and then plantation or natural destruction. And to the knowledge of the local community about the importance of orchid conservation and the lack of knowledge about the various types of orchid has resulted in this potential orchid species being neglected or destroyed because they are considered as a pest, and they may even be new species that have not been identified. And then next, we then think that this through orchid we can support the green economy and also the climate resilience. And next, please. So our research objective, we want to identify and use the natural resources as a funda. Tree color has ecological and economic value, and we want to explain and analyze what and how to leveraging biodiversity, especially tropical wild orchid in Indonesia. And our material and method, we conduct this research during August this year, 2023, in Batur Cangkringan. Why we choose this place? Because we want to bring it up about the local community in Batur village, Cangkringan, Yogyakarta. They are Mr. Sujono, we call it Mr. Jono, and then we interview him, and then we talk about how if we develop this Vanda tricolor to support conservation. Next, please. I'm sorry, it's not so. So this is the important inventory list of tropical orchid in FPL Palem. Why we want to show this data? Because we want to show this is the conservation of orchid that we conduct in a correct manner. We follow the procedure, and then we also have a breeding permit to do next development of this orchid. We have 19 species of orchid there, although they belong to number 2 in CITES status, which is species that are not necessarily now threatened with the extinction, but that may become unless threat is closely controlled. And then our funding was then, we got the literature review from BRIN and from our office, and then we got this density visualization from the, sorry, please, next. And then this is the visualization, not so clear, I'm sorry. So this is the density visualization that we got from literature data, and also from the left side, there we have visualization overlay. From the figure 1 and 2, we then cluster in visualize with a density-based network between keyword correlate with the title. And the bigger the cycle or the note that say the keyword taken to the title, abstract word, it means that it is the novelty. And next slide, please. And we got the word cloud for this orchid, tropical orchid conservation, but the color yellow is the novelty one, but we want to still continue to this orchid conservation. And next, we now discuss about the Vanda tricolor. So Vanda tricolor is one of the wild orchid that endemic, or we know it is from Sleman, Yogyakarta. That's why we want to develop to support orchid conservation here in Yogyakarta. And then these species belong to Orchidaceae, and one of the most species, rich plant families and diversity. And Vanda tricolor is a monopodial orchid, and also the Vanda tricolor flower have white color, and that one is unique one. And as an economic value or orchid to support green economy, we try to conduct a tissue culture technique to develop this Vanda tricolor. And then this Vanda tricolor has high economic value among people who love ornamental plant, or we are in Indonesia, or especially in Yogyakarta, we have club who love orchid. It's called PAI, or it's called like Kagama orchid. And then, therefore, developing this orchid could be profitable business opportunity. But as we know that conserving orchid, especially tropical orchid, is a little difficult. That's why we want to make a collaboration with many of them. And then the conservation status of Vanda tricolor. So Vanda tricolor in Indonesia is spread across various regions, from the East Java, West Java, Yogyakarta, Bali, and then Sulawesi. And this species is natural habitat has become scarce due to natural disaster and human activities, and especially in Yogyakarta in 2010, we have eruption of Mount Merapi, so that's this Vanda tricolor even more difficult to find. So we want to, again, doing conservation to this tropical wild orchid. And as in an observation orchid in FPL Palem that we conduct, so we also observe the big tree around this FPL Palem, and we found this tropical wild orchid could be possible as a carbon sequestration, because orchid can be an alternative carbon sequestration because of their living habitat as well as their respiratory in being able to live as epiphyte. The advantage of carbon absorbing epiphyte orchid include being able to adapt well to minimal light because they need shading, especially filling the surface of the bark, which means increasing the area of CO2 absorption compared to the surface of tree bark without epiphyte orchid. So being adaptable to limited water, nutrition, as well as acting as ornamental plant. Okay, so next we may jump to the conclusion. So Vanda tricolor has high economic value among people who love ornamental plant. That's the first conclusion. And the second one, the results of this study indicate that biodiversity resilience can be formed from the role and function of citizens to have a collective awareness in exploring, caring, and preserving, as well as leveraging of tropical wild orchid. And then the third, the influence of climate on tropical wild orchid will have an impact on their existence, both in their natural habitat and in the greenhouse or nursery where they are cultivated. And last, further research about the potential adaptation to zero stress of tropical wild orchid from the aspect of anatomy and physiology is urgently needed. Thank you very much for your attention.
Thank you so much. Terima kasih Ibu Nura for the interesting presentation about wild orchid, and I didn't know that it was endemic here in Indonesia, in Jogja. Nice. Any comment or question about orchid? Any orchid lover in this room? Anyone from online who would like to ask question? I think there's one message here. A question from Ms. Dian Saptarini. Is the Vanda tricolor an identified flower, or will it be developed as an identity for the Merapi area? Ibu Nura? Apakah itu bunga identitas atau akan dikembangkan sebagai identitas Merapi kayaknya maksudnya ya?
Well, thank you for the question from Mbak Dian Saptarini. So Vanda tricolor is endemic, and we want to develop this orchid, tropical orchid flower. If it could be an identity for the Merapi area, well, we want to support it.
Thank you. So it's not yet kind of officiated as a identified flora of Merapi or something like that?
Yeah, actually, I'm not certain yet because there's a lot more research to do that. I mean, for the development of this flower. So it should be known for the public, but it is not yet. Okay. So I think we support toward that.
Okay, okay. So that's the big homework for us to publish it, yeah?
It is, yes.
Thank you to Ibu Nura. Let's give a round of applause.
Thank you very much.
Next, we will have Miss Avritania Sabila Putri. Do we have Miss Afrita here in this room? Okay. She will be presenting a talk entitled Temporal Genetic Diversity of Sandalwood in Giring Landrace at the borderline of southern and middle zones of Gunung Sewu. To Miss Avritania, the time is yours.
Thank you very much. I present this result of my paper on behalf of our team. Actually, my name is Yeni. I am the project leader, but we have our team. There are consisted of 3 researchers. We have Avritania over there, that Miss Beautiful, please standing up. Actually, this is the part of her thesis, but she asked me to present. I was her supervisor, actually. Okay. Today we have a result of our research on the temporal genetic diversity of sandalwood in Giring Landrace at the borderline of the southern and middle zones of Gunung Sewu. Okay, next please. Next please. Okay. Why we choose sandalwood? First, I will talk about the extinction status of the sandalwood. Globally, it listed as vulnerable by the IUCN Red List, but in fact, locally it is more than this, since it has more than 90% decrease on the population diversity. So it is critically endangered or even extinct in the wild. Particularly in the eastern parts of Indonesia, which is their origin. Considering this, so the sandalwood in Gunung Sewu could consider to be the alternative sources for the rehabilitation. Sandalwood in Gunung Sewu derive into many land races. But Gunung Sewu itself varied due to the differences on the formation history. So landscape differences reflects to the variation on genetic and population structures of the existing sandalwood ranges. And in Gunung Sewu, the sandalwood ranges are centered in the middle zones. The middle zones derive into two parts in which the first is the Ledok Wonosari formation, which is in the past derived from the alluvial basin or valleys. And the second one is the boundary of the Sambipitu and the Wonosari Punung formation, which in the past was derived from the faults. In Indonesia, we call it patahan. And recently it forms the many underground caves, and also uphill caves and the cliffs. So, okay, next. this is the recap of our long-term research on the sandalwood. Up until now, we have observed 24 sandalwood population and land races, which is distributed all over Indonesia. You can see here in Java Island, we have 14, and then 6 in Sumba, and then 4 in Timor Island. Okay, next. So I will emphasize here why in the middle zone is important, particularly in the borderline, the borderline between the Sambipitu and Wonosari Punung formation, because in this landscapes, in the past it happened from the up and hill formation. Up and hill formation, which forms the falls or patahan in Indonesia. So it consists, recently it consists of the luang caves and the cliffs. So we can see here, this formation consisted of 3 sandalwood land races, but the other 2, the first 2 are already degenerated. The first we can see the Behaviour land race. In the past, it happened to be in the high genetic diversity of the parents, but nowadays, due to the illegal harvest and the conversion into the ecotourism activities, so the sandalwood land race become degraded, and it only have very low diversity. And the second one is the mulo. It has also has the degenerated land race, but due to a different scenario. In mulo, the landscape is cliffs, so this is the isolated one. So due to its isolated condition and limited gene flow, and it undergone the gene flow barriers, pollination disruption, lack of seed pads, and reproductive failure. So here the parental diversity is still high, but no seedling found, no regeneration found due to a lack of seed pads. So due to this reason, this Giring land race and the third land race is of importance. But we have no or little information in this third land race, so this research is, we have to conduct this research. Okay, next. So, considering that the Beziarjo and Mulo land races suffer from limited gene flow, decreased genetic diversity, and reproductive disturbance, so the presence of other races in this zone is of importance to be a reservation zone. Giring land race is expected to retain alleles and genes that have been missing from the other 2 races. And therefore, this study aims to determine the temporal genetic diversity of sandalwood in the flowering period of 2022 in Giring land race. Okay, next. This is the study says in the map number 15 in the number 15 in the map. Okay, next. Next. Yeah. In brief, I can state the procedures. The first, we make the inventory and mapping of sandalwood, both in the parents and seedling levels, and then we estimate the clonality level, measure the flowering parents proportion, and then we conduct the isozyme analysis. Okay, next. Here's the results. We can see here that sandalwood parents grow in clusters, in separated clusters, depending on the santalum depth. So this land race is derived from the hills and caves, and therefore the soil has a different slopes like this. So when these soil are deep, there are many trees with generative reproduction, but in the higher slopes there are few parent trees, and this soil depth also affects the level of clonality.
5 minutes, Ibu.
Okay. Seedlings also grow in clusters, divided into 5 groups, marked in red in these maps, unevenly distributed, and not always grow under the parents, since the seeds are eaten by birds. So usually we can find seeds under the habitat of the birds. Okay, next. So we can see here that only 23% of mature individuals are bearing flowers. Among these flowering trees, 11% are derived from the generative individuals, which has a high genetic diversity, while another 2.9% are clones, which has identical genotypes. Okay, next. Yes. Thin salum and abundant rocks supports the formation of horizontal roots and root suckers, and because of this, the parent reproduces vegetatively and forms many clones, and because of that, we observe the increase on genotypically identical individuals. It may reduce the parental heterozygosity as well as alter mating system to be more inbreeder. Okay, next. But fortunately, from this chart, we can see that this Giring landrace, the below one, can retain the allele that were missing or rare in the other previous landrace. So there are many alleles missing or rare in both Bejiharjo and Mulo, but they are present in the Giring landrace. Okay, next. Okay, and this is frequency. This is quite high. Okay, next. Okay, we can see here that the heterozygosity is high on both seedling and parental levels, and the fixation index is negative, which implies this is that they have the outcrossing mating system. Okay, next. This is the allele frequencies of the flowering individuals, which is also still high. Okay, next.
2 minutes, Ibu.
Okay, okay, next. Yeah, also the flowering parents also still have the heterozygosity, the high heterozygosity and the outcrossing mating system. Okay, next. For the I don't know why the format is different here. Okay, for the conclusions, all alleles that were rare and missing in the other landrace are can still be retained in the Giring landrace. So, and in Giring landrace, the parents still have high genetic diversity and also still have the outcrossing mating systems, both in the flowering one or in mature individual in the total, as well as the seedlings. So the seedlings still also have the high genetic diversity. Okay, next. And here we recommend to make the copy of the parents which have the superior characters. So the meta trees in Giring landrace which can retain the alleles supposed to be propagated both the vegetatively or by collecting the seeds. And individual having raw alleles can be used as the ex situ planting materials or in situ planting materials. Also to make the genetic infusion to the other land race, and also to make the introduction materials. I think this is the end of my presentation. Thank you very much for your attention.
Thank you. Thank you very much to Ibu Yeni and her team for the interesting presentation about the genetic diversity of sandalwood. Yeah. Any comments or questions from the audience? I think this talk really sets the scene about how science in genetic diversity can lead to conservation actions. For example, like when was mentioned on the recommendation about transferring the genetic to the least diverse area and preserving the rare genetics that are available in other areas. Any other comments or question from the online as well? If not, then thank you to Ibu Yeni and her team again. Let's give a round of applause. Now we'll move on to the next presenter of today. I think she is attending online. Mega Oktavia Gunawan, are you online?
Yes, I'm online.
Okay, thank you. Mega will present a presentation entitled The Seed Germination of Peat Swarm Plants on Orangutans, my favorite animal, fecal, in Cabang Panti Research Station. So coming up from Sumatra, I think? Oh, Kalimantan ya?
I'm from Kalimantan, yeah.
Okay, silahkan Mega.
Maybe I need a permit, okay. I will share the screen first. Okay. It's already seen?
Yes, you just need to put it on slideshow, please.
Oh, slideshow, okay. I already put it on slideshow. It doesn't.
It's just lagging a bit.
Oh yeah. Everything all right?
Can you try again with your slideshow?
Okay. Okay, I will try again. It already appears. Not yet.
Yeah, I think it's okay.
Okay.
Yeah.
Okay, first. Wait. Oh yeah. It already appears.
Ya, tapi belum slideshow kalau bisa di F5.
Oh, okay. Okay, first.
Slideshow atas tab.
Yeah, I already slideshow, but it's not.
Okay, I think that's fine. We can see
fine.
your presentation. Okay.
Okay. Okay, first, thank you for the opportunity. So it's our pleasure to be here. And we also would like to declare that we are free from any fecal fragrance. So it will be great if we also tell the story behind of this research. Why should be feces? Why should be the seeds? So the first is begin from the, oh, sorry. Yeah, I have trouble here. Does it appear? Oh, yes.
Yes, we can see the the title slide still.
Yeah, it doesn't work. It's already moved.
No, it moved. Yeah.
Oh, okay. So it's all begun from the peat swamp forest ecosystem, which is we all know they are very sensitive. They are easily burned, like in the Borneo. So currently they are burned a lot of hotspot in wildfire in here. So the establishment of the peat swamp forest ecosystem is also supported by the substantial interaction between the animals and plants. which is one of them was the seed dispersers. So the one of the seed dispersers that currently being the global concern was the orangutans, and they could disperse the seeds far away from the mother plants to the new location where they defecate. And there is a lot of literature that said earlier germination could support the species to grow, or the least competition between the other species. So our aims are one would like to find and analyze the difference of the temporal seed germination that distribute at Cabang Panti Research Station. But we had to find how to get the results. So we carried out our research at the Cabang Panti Research Station, which is in the Borneo Island, and specifically in the Gunung Palung National Park. And we built our research in December 2022 until the April 2023. And the timeline of this research was, first we built a sampling, which used vocal animal sampling to collect the behavior data of the orangutan, so we could validate what is the feed plants that they consume during the follows. And we also use purposive sampling to collect the fecal samples from the orangutans, so we could gain seeds from the fecals. And germination test was conducted for 45 days with 3 treatments, which is they are consist of the fecal seeds, manual extraction, and whole fruit. And the data analysis we use consists of two data analysis, which is they are descriptive and inferential. And we use our RStudio and Microsoft Excel to tabulate our data. And our significant levels is a list of the 5%. And the result was questioning about what is the content seed species on the orangutan fecal. So we find that 40 of 54 recorded defecation, we identified that 27 samples contain seed, or are approximately 72.5%. And 72.5% that collected from the adult and adult skull and female, which is their 80%. And the least collected sample was the juvenile and infant, which is 2.5%. That is very little or very least samples because they couldn't make a metabolism while they still being the infant. And if we go back, and here we also have found that 7 out of 16 defecate intact seed was coming from the 16 species or 13 families. So orangutans usually handling the seeds or seed handling projection with swell load, and the second was predate, and the third was beat. So the swell load one is well, the seeds have a little or a small like properties, and the species habitats of these all intact seeds was not intact seeds, but the all species that consumed by the orangutans was 75% trees and 25 liana. And the frequency of occurrence that consists on the fecals of orangutans was dominantly by the ficus sp, or 85%. And the most least was Dracontomelonta, which is we only found one seed in the fecal. And the seed property that we measured, we try to compare between the length, the width, the thickness, and the mass. So we found that although in the Tarsis research found Tetramerista glabra was smaller than Cabang Panti Research Station, they couldn't avoid predation from the orangutans, which is they still damage in the faces of orangutans. They also we also have reviewed literature that Vogel have explain that orangutans usually will give more efforts to eat the fruits, which is they are usually unripe, and it's more tough than the ripe fruit. So usually that happens to the Ponga pygmaeus fulbii than the Ponga abeli. And is there any temporal germination between the treatments? So we would like to try index the seed phase first, which is we found that a lot of the species or the treatments have that. So it's very dramatically like it's hard to find that why they are that or why just a little alive in the seeds. So we found that Tutil have explained that the dead seed is usually limited by the environmental factors, so they not easily to be germinated. But we also find that a lot of predation between the seeds, which is you could see the red holes in here for the Microcosanthe desmifolia. There is a hole in the mesocarp, so usually the ants coming and eat the mesocarp to create a hole so they could eat the embryos. It also happened to the Tetramerisda glabra and Garcinia parvifolia. And the whole fruit usually attacked by the fungus. So the fungus is usually grow in the humidity. So that's attack the mesocarp of the fruits. So actually this is so actually wait. So actually we have temporal seed germination between the treatments of Microcosanthe desmifolia because the Tetramerisda glabra doesn't appear or doesn't germinate. So it's only attached to the Microcosanthe desmifolia, which is there is no significant level of significant result between the treatments, but they showed significant in the proximitas, which is habitat is less than 5%. And the fecal seed germinated on the days between the 16 to the 23, and the later they continue to germinate in the day of the 44. And it raised by the manual extraction seed germinated on the 40 until the 44 days.
5 minutes.
Oh yeah, thank you. So we have found that Ganesan that explain if the ecology of the Microcosanthe desmifolia is in the freshwater and lowland to the hill, dipteral carp, and secondary forest, which is we found the Microcosanthe desmifolia in the freshwater, but we don't get the coordinate of the Microcosanthe desmifolia in the peat swamp. So we assume that Microcosanthe desmifolia is also available in the lowland forest. And our result in the generalized linear mixed model have showed no significance. So GLM for both in the habitat and canopy density showed less than 5%. And the canopy density are in between 94.45% and the least is 82.84%, which is number of the germination even in the peat swamp forest ecosystem is higher than or almost equal with the freshwater swamp. In conclusion, we have for conclusion, which is orangutans disperse intact seeds 7 of 16 species, and only Coxmitas showed significant result. Visually, fecal seeds are germinated earlier, and predators who cause the seed mortality were assumed mammals and insects. We also would like to thank you to the Gunung Palung National Park Behaviour, Yayasan Palung, and University of Tanjung Pura that support our findings. We also have special thanks to the field researcher that help us collectively in the field because the conservation is not only work standing alone. Thank you very much.
Thank you to Mega for the presentation. We have 2 minutes maybe for discussion, Q&A. Anybody would like to give some comments or ask any question? Again, I encourage participants online to also, Mark raise his hand unless anyone here. Okay, can I go ahead, Mark?
Thank you. I was just a bit curious. You had the slide on the germination index of seed fate for different species, and you had the table, table there.
Try to share the screen first.
Yeah, thank you.
Yeah, the seed fate. Where the seed fate? Okay. It appears or
this one, this one. Maybe I just misunderstand. But like for the first one there is Garcinia bancana, and you have treatment A. You have treatment A for that, and 0% germinated, but 53% are alive. And I don't understand how that is possible. Because if they didn't germinate, then they are not alive. I don't understand. Could you please clarify that?
Yeah, thank you for the question. So, while we during the observation, we try to collect the Garcinia bancana seeds. So we divided the categories as the germinated, alive, and dead. Which is the dead we brave to say it that because the cages. The seeds is lost from the cages, so we assume that the seeds is broke by the other animals, and alive is still in the cages, which is they are still intact, and there is no broken in the seeds.
Okay, yeah, I understand. Thank you.
Yeah, you're welcome.
Thank you. Yeah, thank you to presenters. Let's give a round of applause for the presentations. We need to move on a bit faster here because we still have 3 presenters. May I kindly request Dr. Dian Saptarini, she's online, to give her presentation entitled Diversity and Conservation Status of Avifauna in Sampung Karst, Ponorogo. Dr. Dian, are you with us?
Yeah.
Okay, good. The time is yours, please.
Okay, thank you very much. I already sent my presentation to the committee.
Yeah, I think they're uploading it.
Okay, thank you. Good afternoon. Good afternoon to all of the participants of ISWBC 2023. This afternoon, I would like to present our research about the diversity and conservation status of avifauna or birds in Sampung Karst, Ponorogo. Actually, we still waiting for the.
Okay, I think there's a bit of technical issue with the computer here.
It doesn't matter. Thank you. Congratulations for the committee that have the hybrid symposium very great.
Yes, actually, let's give a round of applause to our committee here today. Kasih semangat.
Semangat sore.
Silakan Ibu Dian, sepertinya sudah.
I will try. Let's see. But I still in my. Is it already changed to my presentation or still the previous?
Actually, on the screen in front of us, we can see your presentation, but you're right that on Zoom, it is still the previous one.
Okay.
So let's give a few seconds for the committee to set that up.
Okay. Okay, I will try to introduce myself. My name is Dian Saptarini, and we are a team in my laboratory, ecology laboratory from Institute Teknologi 10 November from the biology department. Why we try to present about the bird in Sampung Kars, Ponorogo. It is related with the plan for the government or local government, Ponorogo, to develop the Sampung area, the Kars area in Ponorogo as an ecotourism. Okay, next. Please, nah, this is Sampung Hill with the in the red area. The Sampung Hill located in Ponorogo district that dominated with the limestone area. There are several ecosystems in the Sampung area, but it is dominated by thick forest, belongs to Perhutani Madiun. And another ecosystem in Sampung area is about like a rice field and the settlement area. And also in Sampung area, we can find the mining activities. Nah, the unique characteristic of the Sampung Hill make this area to be utilized as a tourism destination. The local government has the plan to develop the area, become a rock museum. And in this area, there are so many birds or avifauna that use the Sampung area as their habitat. Now, related with the activities, we try to identify the composition and the diversity of the birds in the Sampung area. And the result of the study, we hope can be considered for the local government to create the master plan for Sampung as an ecotourism. Next, please. Okay. This is the material and methods for our research. The technical data for avifauna collected by the point count method, where the researcher make a plot 10 meter for vegetation, and the vegetation diameter of sample by categories of tree and pole with the diameter more than 10 centimeter. There are 11 observation stations based on categories of thick plantation. We have station 1, 3, and 4, and newly planted thick plantation, and station 2, 5, and 6, and 8, thick plantation with mixed vegetation in station 6, 9, and 11, and mixed vegetation with grassland in station 10 and 7. Based on these categories, we will relate the condition of the habitat with the diversity of the bird. Okay. For the birds data, we will analyze the diversity using the Shannon-Wiener index. And interrelationship between the tick plantation habitat, and bird community was investigated using the multivariate analysis. Next, please. This is the map of the land cover of the vegetation in the Sampung Hill based on the diversity and the habitat of the tick. We can categorize or group the plantation become 4 group A, B, C, and D with the differences 3 species, 3 abundance, and also 3 diversity and the 3 diameter in the 4 group. Okay. The tick plantation in the group A has the highest average tree abundance. Oh, sorry, the newly plantation because of the small size of the tree, I think. Tick plantation categorized based on the tree diameter shown the significant differences. It means each of this tick plantation has different characteristic of the type of vegetation respect to the age of planting. Okay. So we can differentiate the A group and B group based on the age of planting. The A group is the existing tick plantation, and the B is the newly plantation or in the rehabilitation area. Next, please. Okay, this is the diversity of the birds in the Sampung Hill, species richness. We can find 42 species bird in the Sampung Hill with an average of 12 until 28 species at each station. And totally 843 individuals identified in the Sampung Hill. The family Astrilidae has the highest numbers of individuals among the recognized species, with an average of the Astrilidae 172 individuals. We can see the proportional of each family in the Sampung Hill in a pie diagram, and it shows that habitat characteristic in each sampling location will be give an influence of the different composition of the bird species. This is the birds that always found and have a high abundance. Ini adalah lonkura punctulata, Pycnonotus aurigaster, Colocalia linci atau walet, and Geopelia striata. Next, please. Okay. From the 11 stations, we got 3 stations are mostly used by birds are station 9, 10, and 11, with the species richness ranging from 19 to 28 species. These 3 locations also have the high standard winner index diversity with the value range from the 2.68 to 2.99 or in the good category. Yeah. This is the abundance in the blue bar, species richness in the orange bar, and the biodiversity index from standard winner in the point in the top of the graphic. We can see the differences species richness and also the abundance of the birds in every station. Bird species composition, why it is different from the station 1 to until the station 11. Bird species composition is a response of the disturbance and on competition in their habitats. We can relate the abundance of its station with the tree species. Forest area with forest area with planted land cover types tend to rarely disturbed by human activities, so that birds use them as habitat. Station 9, 10, and 11 covered with thick plantation, with mixed vegetation. And this is far from the human settlement. So in the 9, 10, and 11, we can see the abundance and the richness of the birds higher than the other station. Next, please. This is the, cannot see the clearly. I will try to see. This is the species checklist. Okay, this is the species composition in the 4 habitat types. The thick plantation, okay. In my computer, the mixed vegetation and mixed grass. Okay. The species composition in the planting area consists of the several species that are only found in area with the certain habitat, namely Megalima hymocephala, Copsychus sularis, which are associated with the large tree in the forest area. Maybe we can move to the next slide. Next slide. Okay. This is about the vulnerable species that we identified in the Sampung Hill. At the station adjacent to the limestone cliff as a nesting habitat, species specialists use the cliff as their habitat, like Hirundo tahitica, Pada oryzifera, and Falco moluzensis. The species are types of birds that make nests in cliff holes or large tree and are observed heading towards to the cliff area, the limestone hills, in the afternoon. The species deserve attention because the limestone cliff habitat in the plant development area is the main object for the development of the rock museum. So it is clearly has a specific impact on this species, considering the Pada oryzifera or gelatic has endangered based on the International Status Conservation or IUCN. Next slide, please. This is some of the species in the Sampung Hill, some of the bird species in the Sampung Hill that correlated with the Indonesian regulation conservation and IUCN status. We have 7 species: elang tikus, elang ular pido, gelatik Jawa, alap-alap sapi, and betet biasa. Referring to the international protection status of the IUCN Red List, 1 species is endangered, 2 species are near threatened, and 1 species is vulnerable. This presence of this species is a parameter that requires attention, both in terms of saving the species and their habitat condition. Next. This is the conclusion. The different types of vegetation covered in the karst ecosystem of the Sampung Hills have resulted in the formation of various associations of fauna, composition to habitat forms. And the condition is due to fact that some species are highly dependent and associated only with a few habitat conditions, such as feeding ground, nesting ground, and spawning ground. And for our research, we want to thanks to the Institut Teknologi 10 November and Dinas Pariwisata Kabupaten Ponorogo, and also our laboratory, ecology laboratory, for supporting all of the equipment during the research. Thank you very much.
Thank you very much, Dr. Dian, and let's give a round of applause for the presentation about ecosystem and how it connects between animal behavior and the ecology. Unfortunately, we don't have time for a discussion, so if you would allow me to continue to the next presenter, but again, thank you, Dr. Dian. Right, the next talk will be from Khairul Ihwan. Do we have him? Okay. He will give a talk about conservation strategic development of wildcats in Central Kalimantan Province, Indonesia. To Mr. Khairul, the time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. My name is Khairul Ihwan. So I am representation from Borneo Nature Foundation. So this is a collaboration between, not between, but among BNF, BKSDA, Central Kalimantan, and also others organizations like both in private and also private sector and also the university. So we bring the story from Central Kalimantan to initiate the working group of wildcats in our working area. The title with Conservation Strategic Development of Wildcats in Central Kalimantan Province. So today they are a representation of BKSDA Central Kalimantan, Pak Mubarak. Hello. Next. So sometimes if we talk about wildlife in Borneo, we will focus on the big mammals like Bornean orangutan, or sometimes rhino, or maybe elephants. But next. There are another important species, for instance, Felidae or wildcats, and why they are important in the wild. Next. Okay, next. Because they have a terrestrial ecological role as a predator, so it can balancing the food change in the wild. The second one species in endemic, a bay cat is endemic in Borneo, and then the third one is threatened. We need to realize that they are facing a lot of issues. For example, like now, there are several points of forest fire in Central Kalimantan. And then the last one is protected. So all species, wildcats in Central Kalimantan, were protected by Indonesian government. So I think, and we considered they are important to make a future strategy for conservation development. So there are five wildcat species. Okay, can you press the button next? Yeah. So the first one is a leopard cat, the most common cats in Central Kalimantan, because we can find this species in different and several habitats. And then there is a flat-headed cat, and then leopard cat or macan dahan. And then on the right side, a marble cat, and then in the center is bay cat. Bay cat is a Catopuma badia, and endemic in Borneo. So sometimes maybe we think this species will feel sad because they are less concerned. I mean, compare with other species in Central Kalimantan. So maybe they are sad because there are a lot of potential research and also developing conservation strategy. And then we think about to gather all the organizations, stakeholders, to discuss about what we are going to do next and how can we assess the actors or the organization who conduct the research, the activity related on wildcats in Central Kalimantan. So next. So the question, how to support wildcats conservation to be happy cat, not a sad cat. Next. We realized that we cannot conduct these activities and initiative alone, so we need to collaborate with all the organizations in Central Kalimantan, of course from the governments, BKSDA Central Kalimantan, National Park, like Sebangau National Park, Tanjung Putih National Park, and then we try together a lot of practitioners in conservation, like RMU, PT SSS, who conduct camera trapping annually, I mean, in their concession areas. And then the university, next. So here we are, Pak Mubarak. He is on the pictures, and we gather all the organizations in Central Kalimantan to collect the information about the wildcats. At the first time, we are going to collect all the information related on wildcats, but there were several suggestions. So because this is a kick-off meeting, maybe they will be shocked if we force them to collect the information about the wildcats information related on camera traps or smart patrol. So we are going to try, next, okay. So the kick-off meeting was held on July 2022. So it's been a long time, almost a year. And then we are still running on to approaching BKSDA and also other organizations to collect the informations and also to establish the working group in Central Kalimantan. So the kickoff meetings split into two different approach. The first one is individual assessment. So we would like to understanding the knowledge level at senior level, because the participant is like a Paulhood or ranger, and then the university lecturers, and also others participants who have conducted activity related on wildcats, and also not only wildcats, but also almost in every conservation activities in Central Kalimantan. And then the second one, we conducted focus group discussions to understand the actors who are the organization who conduct the research or activity related on wildcats, and then the location, so we can understand whether the location for the wildcats research or information or data will be spread out in Central Kalimantan or not. And then the activity, we would like to understand what they have done or what they are going to do for the next years. And then the last one is threats, because yeah, we need to know about the threats, and we will prioritize how to support the conservation of wildcats in Central Kalimantan. Oh, next. Yeah. So there are a small number of participants, only 25 participants within 12 organizations. But we know from the kickoff meeting, there are diverse participants from the early stuff. I mean, the responding working experience from 0 to 10 years, 11 to 20 years, and also 21 to 30 years. So it's a combination and the diverse participants of the senior level in Central Kalimantan who conduct the conservation activity related on wildcats. Okay, next. So we conducted a participatory mapping, so we can understand where they conduct the activities, and we can understand which activities they have been conducted. Next. And then this is a focus group discussion, so we can understand the threats, so we can range from the most threatened and then the least. Next. So this is the result. So at the knowledge level of a senior level, relatively low. Okay, thank you. So we can understand only kubuk or leopard cat, they recognize at least 60% of participants. And then the rest, more than 50%, and then the less is less from 50%. Next. So we can understand the actors, the organizations and their locations. So it will be easily to collect the information related on wildcats, because in this year we are going to conduct the follow-up kickoff meeting. So I hope it will be initiate the working group in Central Kalimantan, led by BKSDA Central Kalimantan, Pak Mubarak. Next. And then we can understand the activity they have been conducted. There are camera trap, rescuing, patrolling, research, both inside protection area and outside protection area. So we can understand not all the organization conducted activity related on wildcats, like API, Kalteng, and also Dinas Kehutanan Kalimantan Tengah. Okay, next. So this is the threats of wildcats in Central Kalimantan, both in outside protection area and inside protection area. The most threatened is hunting or poaching, but we do not have any information yet until now. The reason, whether this is for fulfill the necessity or just for hobbies. Next. Okay, this is a summary. So there are 4 different activities in Central Kalimantan related on wildcats. So we know the actors and the activities. And then the second one is knowledge on wildcats at senior level is still low. So we need to give more awareness about the wildcats in Central Kalimantan with the help with BKSDA Central of Kalimantan. And then the FGD identified the main threats of wildcats in the province is hunting. Next. And we are going to follow the information related on our kickoff meeting. The first one is approaching unlisted or not coming organization, because there are several organizations we have been invited, and then they're not coming, didn't coming. And then second one, establish working group. And then this is the change for us. If we have conducted, established the working group, we can support the land-based assessment of leopard cat in Central Kalimantan. Not only Central Kalimantan, but also in Indonesia. But in Central Kalimantan, will be led by BKSDA. And then we are going to conduct a social survey to reveal the reason of hunting, whether it is for the fulfill their necessity or just hobbies. Okay, next. Okay, thank you for your attention. And this is, I appreciate with BKSDA of Central Kalimantan, Pak Mubarak, within help hosting and also organize all the things about the kickoff meeting and also the follow-up plans. Thank you so much.
Thank you, Mr. Hairul, for the presentation. Anybody would like to give comments or questions? Silakan Ibu. Ada mic.
Thank you. My name is Elin. I'm independent consultant, have been working with communities for the past 5 years, especially the social forestry communities. So in the list of the stakeholders, I didn't see the community stakeholders. But in the follow-up plans, I'm quite happy that the third part is social survey. But I think probably it's better not only social survey, but you can include them into the developing the plans too. If you would like to know more about this, please come to the Symposium 3 tomorrow. Yeah. I'd like to talk about a little about this. Thank you.
Thank you. We realized about it, and after the after the kickoff meeting, we realized that there are no community involved in our kickoff meeting. So we need to follow up again, not only for the stakeholders, the university, the private sector, or the palm oil plantation in Central Kalimantan. But so we are going to invite LPHD. Yeah. Thank you.
Okay, thank you very much, Mr. Hairul,
thank you
for the talk, and glad to see that some seeds of collaboration is coming up here. Last but not least, we'll have our last speaker of the day, which is Iksan Fikronau Valianto. Do we have him here? Yeah, he will give a talk about the role of urban landscape design on wildlife biodiversity in Yogyakarta, Indonesia. Silakan.
Good afternoon, everyone. In here, I will present my research. It's about the role of urban landscape design on the bird diversity in Daerah Istimewa Yogyakarta Province, Indonesia. Next. Yeah, we will start with the introduction. Both tangible and artificial landscape factors are part of the generally visible landscape and provide diverse ecological system. Spatial factors of a landscape are naturally composed and formed by environmental change that occur in that location. And so you will see that human activities are one of the main factors that significantly impact of changing natural cover to artificial and making landscapes more straightforward and threatening flora and fauna species. Cities are not landscape deprived of plant and animals, but new place home to unique plant and animal communities. In many urban areas, there is generally one or more large open and green space that are parent habitat to conserve biodiversity. Urbanization tends to increase globally and sometimes cause irregular land use around urban areas. So this study was conducted to find out how urban landscape affect the structure of bird community through environmental and anthropological condition. In general, only certain species are able to survive in urban ecosystem, and how urban landscape affect the structure of bird communities through environmental and anthropological condition. Next. So we will start with the method and analyze. This research was conducted in the province of Daerah Istimewa Yogyakarta, Indonesia, in geographically. I use supervised classification for the quantification of the study area, which is use the Quantum GIS. It's open source. And then the bird survey was carried out in August 2021, precisely at 5:30 to 10 a.m. and 3 p.m. until 7:30 p.m. local time in Yogyakarta and its surrounding. Its urban classification, you will see that is distinguished based on the percentage of the built-up area using the approach which was suggested by John Marslouf in his book, The Historical of Urban Bird Diversity. The classification process using QGIS desktop and with semi-automatic classification plugin tools and supervised classification methods. In here we generate 300 point counts, which is it's distinguished by 3 different urbanization class. Next. This is study area quantification. We will start with the Landsat imagery, and then next with the radio correction metric, and then we will generate supervised classification, then urban classification. This is how we built the supervised classification and quantified the study area, which is make the stratified random sampling combined with the systematic sampling. Next. The result of this study was there were 5 feeding guilds based on recorded bird species in this research, which is granivorous, insectivorous, omnivorous, carnivorous, and nectarivorous. Based on the recorded data, the total abundance of individual birds in the study area reached 7,303 individuals. The highest abundance of individual birds was found in suburban areas, which is 2,995 individuals, followed by urban areas, 2,259 individuals, and rural areas, which is 249 individuals. The abundance and densities of bird in urban ecosystem was high rather than in non-urban habitat generally. Species like javan munia and Eurasian tree sparrow were the species that dominate almost in every level of urbanization in this research. In the aspect of bird species richness, you will see that rural and suburban are relatively has the most species richness. Many bird species have adapted to the, this is because many bird species have adapted to the regional land use change, such as agricultural land, which is in the rural and suburban in Yogyakarta, was dominated by the agricultural land. Agricultural land can be a habitat for bird because it can provide habitat for several bird species. Bird communities can be separate from environmental factors in their habitat based on the result of its regression model. Canopy and shrub cover were positively correlated with bird species richness. But the interesting thing in here, because in urban areas, there was organic waste that affect the bird species richness. This environmental habits in organic waste can be source of food. Birds in urban areas, organic waste can be a source of food. In addition, organic waste can attract certain species of birds such as house sparrow or Eurasian tree sparrow. Therefore, a waste disposal system in urban areas can have a particular impact on saving bird communities. Next. The conclusion is about this study is the study proved that its level of urbanization has a varied impact on the composition of existing bird species. Rural and suburban areas have relatively the exact composition of bird species composition. In terms of the abundance of individuals, the suburban area has the highest bird species richness. This can be caused by biotic and physical restriction on the edge of the suburban area. In addition, the edge of the modified are generally has a higher quality of habitat than the core. However, the highest diversity is found in urban areas. This phenomenon occurs because of the heterogeneous landscape or habitat in the area. This research result proved that urbanized areas can become areas that can support the existence of birds. Anthropological aspects are not only found in urban area but also extend to suburban rural areas. One form of anthropological aspect that dominates suburban rural areas in general is agriculture. The suburban rural area in the city of Yogyakarta was dominated by agricultural areas. Intensive farming systems certainly have a negative impact on the existence of birds through the loss of woody vegetation. Next. The recommendation for this research was first we can conduct a monitoring in the study area, and then we can do vegetation enrichment because it attracts birds and it will support the existence of birds in the anthropological area. And then we can do a research in different seasons because this research was conducted in just a single season in dry season, and then it has a potential of improvement in the gathering of the data through the citizen science, which is we know that in Indonesia has some program that we call Burungnesia. As we can see that that was one of the example of the citizen science program. And then agroforestry system. The reason of the agroforestry system is recommended in here because we see that in the study area specifically in rural and suburban areas that has homogeneous vegetation, it results with just a dominant species, not support the richness, but just a dominant species. And then, thank you.
Thank you very much to Mr. Iksan for his presentation. Let's give a round of applause. I do wish that this research you will continue it to a practical program for an NGO in Jogja.
Yes,
yeah,
I think
good,
so.
Good. Yeah, great. Any comments or questions for this local issue that I think all of us, most of us who are in Jogja can be involved in this project? And yeah, silakan.
Good afternoon. My name is Riri, and I'm from Indonesia. So, since we were mentioned, I'm just curious on how you're going to use our application, your recommendation for that program. Like, what can we help exactly?
Okay, I will answer.
Thank you.
First of all, Burungnesia is Burungnesia is one of the application that I and my colleagues use in the field because it provides a comprehensive data of the bird in across all most across in Indonesia, especially in Java. It's so complete. The data is so complete. If we honestly, I didn't even know that if my research is helped, but I have some hope that my research can be one of the result that the proof that if, the urban areas can support the bird, but we have some threshold that we can tackle that in example that we must keep the vegetation cover that in urban areas that vegetation cover that if we didn't had the vegetation cover in there that was nothing. I suggest that if the question about my data support the citizen science of the Burungnesia itself, honestly, so far I didn't even to think about it about that. But if any chance, I will go for that. Thank you.
Okay. Thank you very much. Another seat of collaboration I see here between presenters and participants. So let's give a round of applause to Isan. And then that concludes our symposia for this afternoon. After this, I'll give it to the MC for further announcement. But as your chairperson, thank you very much for your active contribution to this symposia. I do hope we get some useful information, new seats of collaboration, and just enlarge our network here. So thank you very much. I'll give it to the MC.
All right, then we thank you, Dr. Francisca, for your incredible assistance within the valuable lecture and session, and also all the speakers here with the most wonderful research, and hopefully it will be utilized and also could be implemented further. I hope you are hydrated as well, since like we are going to attend all the session for further 3 days. And after this, we have a further announcement from my partner here.
Okay, thank you, Afif. Once again, thank you to our remarkable speakers, our chairperson, and our fellow conservation enthusiasts for your spirit in this abstract presentation session. After this, we would like to invite you for a coffee break, about 15 minutes. We will continue the next session, which is the keynote presentation with the theme Biodiversity, Conservation, and Climate Change Mitigation and Adaptation at 4:10 You are welcome to join us again in Arjuna 1 or switch rooms to Arjuna 2, whichever you please. Thank you.
Thank you so much, ladies and gentlemen. You could have a coffee break outside, and we'll be back here again on the time that which mentioned by Sabila. Thank you so much.